Hello guys, today we started reviewing section 3 in chapter 8, which is talking about gene expression and regulation. We said that gene expression and expression happens during the transcription process, where while the DNA is converted, genes on the DNA or the instructions of the DNA is converted to a strand of mRNA. In this section, we are going to review how this gene expression is regulated, and it's different in prokaryotic and eukaryotic cells. We said that in prokaryotic cells, turn genes on and off by controlling transcription. We have a promoter and operator, which are DNA segments, part of the DNA, and an operon, which includes a promoter and operator, and one or more structural genes that code for all proteins needed. This is an example of the operon that acts like a switch in the prokaryotic cells, which is called lac operon. This is specific for the expression of the lactase gene to break down lactose. Now we said that the lac operon is switched off when lactose is not present because there is no need for this protein or enzyme to break down lactose because lactose is not present. While when the lactose is present in the bacteria, the lac operon is switched on to make this protein, which is an enzyme, lactase, to break down this lactose. And as you see here, when it's switched off, the repressor protein will be linked to an operator and preventing RNA polymerase and blocking it from transcription process. While when lactose is present, the repressor will link or join to a lactose, and this will allow the RNA polymerase to transcribe the protein. In the in the eukaryotic cells, some things are different because this gene expression is regulated by or controlled by DNA, regulatory DNA sequences, and protein transcription factors, which are the enhancer, promoter, and transcription factors, which um, uh, make the enhancer closer to the promoter and allow RNA polymerase to transcribe the gene. We said that RNA during transcription of the RNA, not during, after transcription of RNA should go through a processing in which RNA includes three major steps. mRNA processing includes three major steps, which are removing of introns and splicing the exons and adding cap and tail. Now the mRNA process that is it's ready to take this message to out to the cytoplasm to be translated into a protein. The exons are nucleotide signals that code for parts of protein, while introns are nucleotides that don't code for these parts. They don't have genes that code for protein. Okay, this is for section 6. Now let's go to section 7, which is the last section in chapter 8, which is talking about mutation. Okay, so we learned before that mutation is changing in the DNA sequence, and mutations may or may not affect the phenotype. We have two types of mutations, single gene and interchromosome, or we can call it chromosomal mutation. A single gene mutation, it is either a point mutation where mutation in which a nucleotide substitutes for another nucleotide. And the other type of single gene is the frame shifting mutation, where some nucleotides are inserted or deleted, and that causes shifting in the codons of the amino acid. Okay, and the other type of mutations is the chromosomal mutation, where it occurs during meiosis. We have two types of chromosomal mutation, which is gene replication, where it happens when homologous chromosomes exchange DNA during crossing over, and some chromosomes will have replicated genes where the other homologous chromosome will have no copy of the gene or the gene. The other type of chromosomal mutation is the translocation, which results from the exchange of DNA segments between non-homologous chromosomes, like in this example. So we learned also that mutations may or may not affect phenotype, such as the one which affect the phenotype is the chromosomal mutations, which have a big effect on organisms. Okay, because there is a break, maybe a break in a gene, or this may cause the gene no longer work. Some gene mutations change phenotype, such as mutation that cause premature stop codon during the expression of the gene. Mutation may change protein shape, and if this protein is an enzyme, the enzyme will not function properly. And some mutations occur at non-coding regions. These non-coding regions, they have regulation for the expression, and this will change the gene expression regulation. Okay, some other mutations that don't affect the phenotype if the mutation is a substitution and have no effect, especially for those occurring in the third nucleotide of a codon, like in this codon and this codon, they both code for the same amino acid, which is lysine. Mutation may occur also in a non-coding region such introns. Okay, and this don't affect the phenotype. And sometimes mutation may not affect protein folding or the active site of the enzyme, and it doesn't affect its function as well. We said that mutations in body cells don't affect offspring, it just affects the person itself or the organism itself. Why? While mutations in sex cells can be harmful or beneficial sorry, to offspring. Now, there are several factors that can cause a mutation, such as duplication error that can happens occasionally, and these duplication errors during the DNA coupling cause mutations. Other factors such as mutagens. Mutagens are agents like UV light, the ultraviolet light from the sun rays, and chemicals can cause mutation. Alright, so these are the most important parts in this section. Now let's start with chapter 9, section 1. In this chapter, we are going to talk about the bio, uh, biotechnology and how the biotechnology relies on cutting DNA at specific places. The first section is talking about manipulating DNA or changing DNA, and the main concept for this technique or biotechnology techniques are cutting DNA at specific places. DNA is very large molecule, but it's still very small to see. Okay, and you cannot pick it up, so you must be able to work with DNA without being able to see or handle it directly. Okay, so what they use some molecular scissors uh, that are called restriction enzymes, and they get these enzymes from the bacteria. Bacteria have these enzymes. These enzymes are called restriction enzymes. They are used to cut the DNA, okay, and slice the DNA apart, okay, to make it easier to study this DNA or make any change in it. We have here also, we said that restriction enzymes act as molecular scissors. They come from various types of bacteria, and these enzymes allows scientists to more easily study and manipulate genes. Okay, okay. So some restriction enzymes cut at three restriction sites, and some restriction enzymes cut at more restriction sites. And this will fragment the air to many fragments with different sizes. some of these fragments have straight across, cut straight across and leave blunt ends, and some cuts are staggered where they leave sticky ends, and these are more useful. Okay, now after cutting the DNA, restriction in maps show the length of DNA fragments, and these are made by the using of gel electrophoresis, a technique to separate DNA fragments. This technique based on using an electrical current which pulls DNA fragments through a gel, and the length of DNA fragments can be estimated from the distance it travels through the gel at certain time. The fragments with big size or large size will travel less distances than the fragments with smaller size, okay? Because they will be faster, the smaller size. They will travel more distance. Okay, this is for this section. Let's start with the last section, which is PCR. We said that there are many techniques used in biotechnology to manipulate the DNA. One of these techniques is the PCR, or copying DNA, okay, outside the cell. This is an artificial technique. This PCR stands for polymerase chain reaction, PCR, and this technique rapidly copies segments of DNA. Okay, we said that some forensic scientists use DNA from cells in single hair as a crime scene to identify a criminal, and they compare the DNA from different species to determine how closely the species are related. So the original amount of DNA is too small, okay, to study. So they take these DNA samples and increase them or amplify it by using the PCR, make many copies of the PCR. PCR is very similar to replication, but PCR is done in the lab. It makes many copies of specific gene, and it's similar to replication. The difference between PCR and replication that replication happens inside the cell normally and one time for each cell. But the PCR, they take the happens when they take the DNA from a cell, and they use some materials to make a copy of this DNA. Okay, and this will help in copying the PCR with the best number of DNA each cycle, okay, and make many copies, and this will make it easier to study the DNA samples. Okay, thank you for listening, guys, and we'll continue tomorrow.